So, my name is Stefanik Sobotkova, and I'm a PhD student from the group of structural biology, which is placed at CETEC, Maastricht University. And today I will present you results from my work, which is focused on structure and infection mechanism of phage plus 19. Some of you might ask, what is a phage? Phages are viruses which infect bacteria. There are different types of phages. We have filamentous phages, and we have tailless phages, which are only composed of the icosahedral head that contains the genome of the phage. There are also tailed phages, also different types. There are phages with contractile tail, there are phages with very long flexible tail, and short-tailed phages. The tail of the phage mainly serves for the genome delivery into the host cell. The phage LUZ19 belongs to the last group of short-tailed phages. Host of the phage LUZ19 is an important human pathogen, Pseudomonas aeruginosa. This pathogen causes severe respiratory infections, and most of the species are actually antibiotic resistant. Phages undergo so-called lytic cycle. The lytic life cycle starts with the phage attachment to the host surface. Then the phage injects its DNA into the host cell, and it uses the machinery of its host to replicate the genome and synthesize new phage proteins. After that, new virions are assembled inside the cell, and under a certain condition, the cell is lysed, which means that the cell dies, and the phages are released from the cell, and this new phage progeny can again infect their host. We studied the molecular structure of phage LUZ19 using cryo-EM and single particle analysis. Here you can see a micrograph of phage LUZ19, and it's composed of icosahedral head. Maybe you can see the tail and the very flexible long tail fibers. Probably the most prominent feature of the head of the phage is its icosahedral capsid. The icosahedral capsid contains the phage genome, and it's composed of major capsid protein. This is one subunit of the major capsid protein, and its fold is very conserved along all tailed phages. If we combine one subunit of the major capsid protein with 6 others, we will get the asymmetric subunit. The asymmetric subunit is composed of a hexamer of the subunits and one subunit that is facing the penta vertex. And if we take 60 of these asymmetric subunits, we will have the whole icosahedral structure. The phage head is further decorated by head cement and head decoration protein. These two proteins mainly serve for the capsid stabilization. If we take the head and cut it open and get rid of the double-stranded DNA genome, the core complex will be revealed. The core complex is composed of 3 different proteins. We have 4 subunits of cap protein, then 8 subunits of middle core protein, and the portal protein, which is dodecameric. Each of these proteins have a special function during the phage life cycle. The cap and the middle core protein serves mainly to be ejected from the phage head before the genome ejection, and they form a pore in the membrane of the host. This mechanism was seen in a close-related phage T7. In this study, they used cryo-electron tomography to study the early infection. Here you can see that the phage is nicely sitting on the membrane of the host, ready to inject its genome. In the second picture, the genome ejection is already happening, and you can see that there is an extra density appearing in between the membranes of the host. This density actually corresponds to the core proteins that are ejected from the phage head. And after the genome is ejected, you can see that the particle is empty. This structure is disassembled because the membranes of the host need to stay intact for the lytic life cycle to continue. If we move more to this special vertex, the portal protein serves as a connection between the core protein, the phage head, and the phage tail. The first protein of the phage tail is the adapter, which is also dodecameric, and it's followed by the hexameric tail nozzle. The tail is further decorated by 6 long flexible tail fibers, which were not solved using single particle analysis, so I'm showing only the N-terminal part of these proteins. The phage tail forms a channel for the DNA to pass through. If we rotate these models 90 degrees, we will get a better look at the channel. In some places of the tail-nosal protein, you can see that the channel is actually much thinner and the DNA, the DNA cannot pass through. These so-called gates are used for the holding of the DNA before its ejection into the host. I've shown you the whole structure of the phage with 19 Sobotkova single particle analysis. Again, the phage is composed of the icosahedral head, the core complex, and the tail proteins. We also studied the early infection process using cryo-electron microscopy. First, I would need to a little bit more introduce the host. The Pseudomonas aeruginosa has two different surface structures. First is the flagellum, which is an organelle of locomotion, and then there are pili or pills. Especially, I will be talking about type 4 pili. The type 4 pili have different functions. One of them is surface sensing. It also serves for the attachment of the bacteria to the surface, and it plays an important role during the biofilm formation. We think that also these pili are playing an important role during the phage infection, as the phage is attaching to this pili either by its tail nosal or tail fiber proteins, and then it gets closer to the host. There are two different mechanisms, and we don't know which one is the correct one, but either the phage diffuses along the pili to the cell surface, or the phage gets in a close contact with the cell as the pili is contracted by the host cell. Our theory was supported by the images from cryo-electron microscopy. Here you can see nicely the host, the Pseudomonas aeruginosa, which is filled with ribosomes. You can see the flagella of the host, and you can see the pili structures. And on the pili, you can see that the phages are attached. From these pictures, we cannot see if they are attached by the tail nosal or the tail fibers, so that will be our later investigation. Combining the results from the whole structure solved by single particle analysis and the structure of results of early infection from cryo-electron microscopy, I can now conclude the whole mechanism of the phage with 19 early infection. So first, the phage needs to attach to the type 4 pili either by the tail fibers or the tail nosal. When it gets to the close contact with the cell membrane, it needs to penetrate through it. For this, not only the tail nosal will be sufficient, that's why the core proteins are ejected and form a pore in the host membrane. And through this host membrane, through this pore that was made, the genome is ejected to the host cytoplasm, while the rest of the lytic life cycle continues. With this, I would like to thank the whole group of structural biology, especially our former colleague Julia Mironova. She was collecting the dataset for single particle analysis, my consultant Zuzana Cenikova, and my supervisor Pavel Plevka. I would also like to thank the core facility at St. State University for the data collection and pre-processing, and I would like to also thank the organizers for letting me here to introduce my results from my work. And I would like to thank you for your attention, and if you have any more questions, don't be shy to stop me after. Thank you.
Thank you very much.